Hello, my name is Ashley Valentina Schwartz, and I'm a computational science PhD candidate working with Dr. Udo George and Dr. Carolyn Sand. And today I'm talking to you all about our recent research in which we developed a tool called danRerLib for danRerLib library, which is a Python package for Zebrafish Transcriptomics. Zebrafish are an ideal model for transcriptomic analyses in which we use RNA sequencing to evaluate the entire transcriptome and identify things like gene expression changes. And you might be wondering why. Well, zebrafish actually have a really high genetic homology to humans, and they have conserved molecular pathways. So if we were interested in understanding the genes involved in a particular disease, we can investigate this in zebrafish and translate it to humans. Additionally, they have very rapid development, which allows for large-scale screening. This is particularly useful in toxicology, where we are interested in how environmental contaminants affect gene expression and could lead to disease later in life. And with all this being said, zebrafish are a key model organism to investigate human health and need to be supported to do so. To get into the specifics of why we developed danRerLib, I want to talk about a current problem I encountered in my own research studies. In my own research, we investigate the effect of toxicants on molecular outcomes by exposing zebrafish to a common pollutant and assessing the transcriptomic response and using functional enrichment analysis. Functional enrichment analysis is a method in which we can identify biological functions, pathways, or processes that are overrepresented in a set of genes. This is oftentimes by representing the set that is managed in these databases called KEGG or gene ontology. These can help reveal hidden connections, disease associations, and add biological context to the research question we have. A current challenge is that functional enrichment analysis tools that are out there often lack flexibility for zebrafish researchers and may not provide a comprehensive view of all the biological pathways that are out there. There's limited adaptability for gene ID types, incomplete coverage of these pathways, as I mentioned, and they lack the ease of use for non-competing experts. And so there was a need for a tool to address these limitations, and that's where danRerLib came in. danRerLib streamlines gene ID conversions and offers specialized pathway analysis tools for zebrafish data, which enhances the accuracy and efficiency of functional enrichment analysis and transcriptomics as a whole. Some key features of our package is that we have gene ID conversions specifically focused on those managed by the Zebrafish Information Network, which is the zebrafish gene nomenclature. Also, we have support for Ensemble and NCBI, which are other popular gene management databases. We also have gene ortholog mapping, so we have support to map zebrafish genes to human genes, which is particularly important if you know a gene is involved in a particular disease in humans. We can map that to zebrafish and investigate the effect of that gene in whatever research question you have. And we also have support for functional enrichment analysis for KEGG and gene ontology. A key thing here again is that layered with orthology, we can actually investigate those pathways that are annotated for humans and not for zebrafish. So zebrafish don't have a lot of annotations for these pathways. They're obviously less studied than humans, but what we can do is we can use gene orthology to actually investigate things that are known in humans and map them to zebrafish and use that investigation for translation. Additionally, we have comprehensive tutorials and guidelines with extensive documentation on our webpage to support zebrafish researchers through end-to-end analysis. To demonstrate the benefit of danRerLib, we performed a case study on zebrafish transcriptomic dataset in which zebrafish were exposed to a water pollutant called TCPMOH and then ran through the danRerLib pipeline. What I'm showing here is that the top 10 upregulated and the top 10 downregulated pathways for KEGG using zebrafish annotations alone. Now, when we include the orthology mapping that is supported by danRerLib, we actually find additional pathways that we were unable to detect previously. And a key feature is actually those pathways related to disease. So KEGG only supports disease annotations for humans. So by using orthology mapping, we can actually investigate how these chemicals may be leading to disease on that molecular basis. So again, danRerLib provides enhanced biologically relevant insights that may not be evident in zebrafish annotations alone. Where am I going with this in the future? We are going to be building network models for toxicity predictions and utilize danRerLib to streamline this process so we can investigate a wide set of environmental contaminants. And in conclusion, danRerLib supports the investigation of biological pathways that may be altered by chemical exposure, and we support zebrafish researchers through their research by our webpage, as I mentioned earlier. If you're interested, you can scan this QR code to visit. And with that being said, I would like to acknowledge these institutions and organizations for their funding and support, and of course my amazing advisors for all their guidance through this project. And if you would like to learn more, please come chat with me at the poster.